Okay, so we're going to move to pillar 2. We're going to talk about paternal unsilencing approaches. And I present to you now Encoded Therapeutics. We have Dr. Sirika Pillay, who's going to talk to us about ETX-201, a vectorized miRNA-based approach for the potential treatment of Angelman syndrome. Sirika.
Thank you to Alison for the introduction, and I'm very excited today to talk to you about the progress that Encoded has made in developing a potential Angelman treatment. These are my disclosures. So I'll start in telling you a little bit about Encoded Therapeutics. We are a genetic medicines company that's focused on developing durable treatments for CNS disorders. And we do this by utilizing our vector engineering platform, which aims to improve on safety and efficacy profiles of gene therapies by optimizing specificity and potency of transgene expression. Our mature pipeline includes gene therapies for monogenic and common CNS disorders. And our lead program, ETX-101, which is a treatment for Dravet syndrome, is currently being tested in clinical trials in Australia, the US, and the UK, where we have the potential to address the full spectrum of disease symptoms. In addition, ETX-101 has been granted orphan drug designation and rare pediatric disease designation by the FDA and orphan drug designation by the EMA. And so with our Angelman program, we're at the point now where we're nearing the end of our preclinical phase. I'm excited to share more about that. So to tell you a little bit about our approach, at Encoded, we're using a vectorized microRNA approach to potentially treat Angelman. And this makes use of an artificial microRNA that targets UBE3A ATS in order to knock down its expression and thereby upregulate paternal UBE3A. This artificial microRNA is packaged within an adeno-associated virus, or AAV9, and it's intended for a one-time ICV administration. And the potential there is for consistent and durable unsilencing of UBE3A. MicroRNA approaches have shown safety and clinical efficacy in treating CNS disorders, an example of this being Huntington's disease. And in addition, AAV9 has ample experience in the clinic, showing safety and efficacy profiles in the treatment of CNS disorders. So today what I'll do is walk you through our preclinical development path in the development of our vectorized microRNA approach. And I'll start by sharing a little bit about our proof of concept mouse studies, which allowed us to establish that a microRNA approach is capable of treating Angelman syndrome. I'll go on to share about the development of our human candidates, where we used in vitro screening and optimized those candidates in an in vitro setting in order to identify the top candidates from our selection. And those candidates went into a pilot NHP study, which was focused on target engagement to identify a lead candidate. And when I talk about target engagement, which I'll talk about quite a bit in this talk, I'm not only talking about being able to knock down ATS, but more specifically being able to upregulate UBE3A, as that is our ultimate therapeutic target. So the final phase before we can move a candidate into IND-enabling studies is to be able to test that lead target for a lead candidate for safety and target engagement at relevant doses. And today I'm going to present some data from that most recent study. It's just come out, and so I'm very excited to be able to share that with you. So in our in vivo studies, we used a mouse-specific vectorized microRNA to target mouse UBE3A ATS. And here what I'm showing is our molecular data for UBE3A ATS and for UBE3A RNA. And what you'll note is that there are 3 groups on each of these bar graphs. The first in dark blue being our wild-type untreated group, the second being our Angelman animals that were untreated, and the third in green being our Angelman-treated animals with our microRNA. And what you'll note in the ATS graph, which is on the far left, is that there is knockdown of ATS expression when we treat with our microRNA. You'll see in the UB3A graph that we were able to, with that level of knockdown, upregulate UB3A in our dosed group. And the levels of upregulation were around 30% of wild-type levels. We also looked to see if we were upregulating protein. And what you'll note in the far right is our dosed animals have green staining, which is representing our UB3A protein expression. And it seems pretty widespread across the brain, which is what we wanted to be able to see. So the levels of UB3A expression that we induced in our mouse model at the dose that we were testing were able to show rescue of multiple Angelman syndrome phenotypes. And this included body weight, locomotion, and partial correction of motor function. And what this data package collectively was able to do was provide us with clear data to support a microRNA approach being able to treat Angelman syndrome. But what it also did was demonstrated that partial UB3A was capable of correcting disease phenotypes. And this is supported by human genetics data, where we've seen in studies with mosaic Angelman patients that partial UB3A expression can meaningfully impact disease presentation. In the development of our human candidate, we made use of Encoded's engineering platform, and we went through a fairly rigorous and extensive process to identify our lead candidate. This was initiated by in vitro screening of over 120 candidates, and those candidates were bioinformatically designed to have minimal off-target activity, in addition to having 100% sequence homology between NHPs and humans. And that was important so that we would be able to test our human candidate in NHPs. Of those 120-odd candidates, we were able to narrow that list down to 30 generation 1 candidates, which demonstrated significant UB3A upregulation in human iPSC neurons. And we went on to further optimize those candidates, resulting in 6 potent candidates that we were able to test in that pilot NHP study. That study allowed us to show that these candidates that were optimized were in fact improved by as much as 100-fold in potency, which emphasized the importance of being able to really fine-tune your candidate in order to get to the best candidate. And for us, that candidate was ETX201. So we went on to further characterize The first step in that characterization was to understand the on-target activity. And so to do this, we used Angelman patient iPSC-derived neurons, which acted for us as an in vitro disease model. And here, we tested ETX201 over a broad dose range. And what you'll see in the figure is across that dose range, in green, is ATS expression decreasing as we increased dose. This was complemented in the blue by an increase in UB3A. We also looked at UB3A protein across this dose range and found that there was a dose-dependent increase in UB3A protein. Next, we wanted to elucidate the off-target profile of ETX201. This is critical in contributing to understanding the safety profile of our candidate. And so to do this, we used an unbiased whole transcriptomic analysis, where we took ETX201 treated human iPSC-derived neurons and compared them to scramble-treated neurons, where scramble is a control microRNA that doesn't target anything. And ultimately, what we were hoping to do here is essentially identify any genes other than our on-target genes that may be modulated by ETX201. To do this, we conducted a differential expression analysis. And so in this figure, what we're showing on the y-axis is significance, which is the confidence we have that a gene that we've identified is a real gene that is showing differential expression. And on the x-axis, you'll see expression change, where in the left direction of 0, a gene is decreasing in expression, in the right, it's increasing in expression. And so we looked at over 57,000 genes, 79 of which had been predicted to be potential off-targets just based on our microRNA sequence. And of all of those 79 genes, none of them were differentially expressed in our analysis. In fact, the only 2 genes that showed significant differential expression and greater than 2-fold expression difference was our UBE3A ATS gene, which is represented in blue, where it decreased in expression, and paternal UBE3A, which is represented in green. So this emphasized for us that ETX201 was in fact highly specific for its disease target. We went on to test ETX201 more broadly in an NHP study where we assessed safety, biodistribution, and target engagement at different dose levels. This was a 60-day study where we delivered our ETX201 via ICV delivery. And this is a direct delivery into the CSF, which allows us to reach disease-relevant brain regions, including the cortex and the hippocampus. There were 2 animals in every dosed group and 3 in our vehicle group. And you'll note here the color coding, where vehicle is in gray, our first dose, which is our lower dose, is in light blue, and our second dose, which was our higher dose, is in dark blue. And I'll use this color scheme throughout the rest of the slides in the presentation. So when we looked at the safety assessment from this study, what we found was that ETX201 was well tolerated in NHPs with no adverse findings. And specifically, we had no clinical findings, we didn't have any clinical chemistry abnormalities relative to our vehicle animals, and we did not have any adverse microscopic findings. We went on to look at biodistribution, and here I'm showing a brain slab where the green staining is our microRNA expression from ETX201. And what you'll note is we were able to see broad microRNA expression throughout the cortex. In addition, we were able to detect ETX201 microRNA in extracellular vesicles in the CSF and in the serum. Now, extracellular vesicles are these sac-like vesicles. They're naturally occurring, and they're known to transport cytoplasmic cargo between cells. And what you'll see when you look at our CSF data is you'll notice a dose-dependent detection of our microRNA at day 60, which was the end of our study. For our serum data, we were able to look at a number of time points across the study and monitor our microRNA expression throughout the study. And you'll see that we had a peak in our microRNA expression around day 8, and we were able to show sustained levels of that expression throughout the study. Finally, we looked at target engagement, and here I'm showing ATS expression and total UBE3A expression. And you'll note that each of the data points on this slide actually represent one analyzed punch from different regions of the brain of dosed animals. So you'll see with the ATS expression data, we saw significant downregulation of ATS in both doses tested. And this was complemented by upregulation that was dose-dependent in our, when we looked at total UBE3A. And we saw this in both doses, where the highest dose had as much as 20% increase in UBE3A expression in the cortex. Finally, we looked at paternal UBE3A expression, which is really our ultimate target engagement. And here we were excited to see that ETX201 demonstrated dose-dependent, widespread target engagement in disease-relevant brain regions, including the cortex and the hippocampus. We were seeing this at both tested doses. So in summary, our data establishes the therapeutic potential of a durable UBE3A unsilencing approach to treat Angelman syndrome. Our in vivo proof of concept studies showed us that partial UBE3A was capable of correcting multiple disease phenotypes in an Angelman mouse model. And ETX201 shows potent and specific target engagement in vitro, emerging as our top candidate from over 120 candidates that we screened. It is also well tolerated in NHPs with no adverse findings in the study that we've done. And it demonstrated broad biodistribution with microRNA expression throughout the cortex and detectable in the CSF and the serum. ETX201 also demonstrated dose-dependent target engagement across multiple brain regions post-ICV delivery, where we observed knockdown of ATS and upregulation of total UB3A over wild-type levels. In addition, we saw widespread and dose-dependent upregulation of our paternal UB3A in critical brain regions. And so taken together, these data support the further development of ETX201. So I'll end today just saying a big thank you to all of the Encoded team members who have worked really hard to be able to advance this program, and thank you all for your attention.